Hello everyone, my name is Layla Hosseini-Gerami and I'm in Andreas Bender's group at the Centre for Molecular Informatics at the University of Cambridge. And today my poster is in the topic of pathway enrichment, machine learning and causal reasoning analysis to deconvolute potential targets of a small molecule tau aggregation inhibitor. So the deconvolution of the mechanism of action, or MOA, of a compound is very beneficial to the drug discovery process for a variety of reasons, including a discovery of side effects, a rationalization of compound efficacy before clinical trial, and so on. But this remains a challenge. In this work, we combined bio and cheminformatics analyses to understand the MOA of a small molecule tau aggregation inhibitor. Tau aggregation is a key histopathological hallmark of Alzheimer's disease. So by understanding how such a compound works, we could pave the way for future interventions for Alzheimer's disease. So in this work, the methods that we undertook were kind of split into two parts. So the experimental part involved culturing rat cortical neurons, or RCNs. We then split the cultured neurons into two sets. So we have a healthy or seeded. And by seeded, we mean we took human-derived tau and seeded it into the cells to induce tau aggregation. Following this, we treated both the healthy and the seeded cells with the small molecule tau aggregation inhibitor for 3, 7, and 14 days. We then performed RNA sequencing and obtained differentially expressed genes, which were relating to compound treatment of the two cell types. So with differentially expressed genes, we performed two different analyses. The first is pathway enrichment, and the second is causal reasoning, which is inferring upstream protein drivers of transcriptional response. We also performed with the chemical structure of the compound some target prediction to predict which targets are being engaged by the compound. So for the results, so in generally across all the approaches, we found evidence pointing to modulation of processes involved in inflammation and cholesterol homeostasis. So for example, for the pathway enrichment, you can see here, these are the results for the healthy cells at the 3 time points. And there are some pathways here which are relevant to inflammation. So you can see here that interleukin signaling, for example, PI3K signaling, and also at day 14, lots of processes relating to cholesterol metabolism. Such processes were also found with the data derived from the tau-seeded cells, which is very interesting because this shows that it's both relevant to the disease and also is independent of and occurs not occurring due to the tau aggregation inhibition. So as for the causal reasoning, analysis, as I mentioned, this is kind of picking up these upstream protein drivers, which are relevant to the compound's mode of action. So these two sterile regulatory proteins were picked up as upstream protein drivers, and these proteins are involved in the regulation of cholesterol homeostasis. And some other upstream drivers included STAT1 and REL-A, which play a role in inflammation. And again, these drivers were observed in both data derived from the healthy and the tau-seeded cells. So as for the target prediction results, so this is based solely on the chemical structure of the compound. The two highest predicted targets were NF-kappa B subunits and also the Niemann-Pick-C1 protein. So the NF-kappa B subunits are involved in inflammation, and NPC1 here is involved in cholesterol transport. We also found that the nearest neighbor in the machine learning models in the training set had a Tanimoto similarity based on ECCB4 fingerprints of 0.5, and structurally these compounds look very similar. And we found that this nearest neighbor had very good activity against the two highest predicted targets, so this gives some confidence to our results as well. So to conclude, through the analysis of a small molecule tau aggregation inhibitor, its biological and chemical data, we found evidence of perturbation to cholesterol homeostasis and inflammation, which are both very relevant to Alzheimer's disease progression and tau aggregation. And we've made these observations on the pathway, protein target, and signaling protein levels. And this could explain the observed efficacy and its mechanism of action as a tau aggregation inhibitor beyond direct tau binding, as it is found to reduce tau aggregation in vivo. By looking at the agreement between different data modalities and methodologies, we are also able to generate these hypotheses with greater confidence for experimental validation and really provide a good starting point for validating these hypotheses. And I would like to acknowledge my Eli Lilly supervisors, Emma Lang, who's now at GSK, and David Evans, who's now at DeepMind, as well as David Collier and Howard Broughton, my Cambridge supervisor, Andreas Bender, and the entire Bender group, as well as the BBSRC for funding my PhD. And thank you for listening as well. I look forward to hearing your comments and questions, and if you'd like to get in touch after the conference, you can find me. Here is my email and here is my Twitter. Thank you very much.